Hello there, everyone. Let us talk about the therapeutics of trypanosoma as such. Okay, so now regarding the therapeutics that is involved in management of this trypanosomasis, my talk would be involving based on two major type of therapies. The first one, what we call it as arsenic based therapies, and other therapies that include molecules like DMFO, difluoromethyl that is usually given in form of eflorothiene. The second other therapy includes nifurtimox that we are going to talk more in details. In this lecture, we would be talking mostly on arsenic based therapy, how these arsenic based therapies were developed with time, what is the mechanism of action of arsenic based therapies like milarsoprol is one of the drug that is one of the major feature of these arsenic based therapy, how this milarsoprol acts, and how the parasites might have gained resistance to these milarsoprols. Okay, so besides these two popular therapies, there are some of other candidates which are still present in clinical trials. Okay, so say for example, fexinidazole is a compound that is present in phase 3 clinical trial, and another compound SCYX7158 that is also present in phase 3 clinical trial. So we can assume that in upcoming 4 or 5 years we might be having certain more positive new drug candidate molecules in market that can be effectively used for management of this sleeping sickness and trypanosoma based diseases as such. Okay, well now let us first begin our discussion on talking with arsenic based therapies. Okay, so these arsenic based therapies are based on inorganic arsenical compounds. So these compounds were used for nearly last 2500 years, and they are as poisons as they are used as poisons as well as therapeutic agents. So the first evidence as anti-trypanosomasis was given in 1858, that these arsenic based compounds can be used as anti-trypanosomasis. That was proposed back in 1858, and it was David Livingstone that put forth this idea. His study was based on an animal disease called Nagana in Africa. So what he did was that he gave flowers a solution. Flower solution is nothing but a popular tonic that contain 1% potassium arsenate. This flower solution contains potassium as well as you can see, arsenate ions as such. Okay, so in 1902, first laboratory confirmation of the trypanocidal activity of arsenite in infected rodents was proposed. Okay, it was Leveran and Mesnil that put forth the idea back in 1902. So in their study, whatever animal model that they have used, all of the animal died from infection if treatment was discontinued. And even though if the treatment was continued, the same animal model died due to arsenic toxicity. So evidently it is quite clear that these early flower solution based or arsenite based solutions or arsenite based drug formulations were not very effective. So later on, in early 1900s, people came out with atoxyl. So this atoxyl, the structure of atoxyl, as you can see here, is an arsenic acid analog. Arsenic acid analog. Arsenic means an acid which has got arsenic rather than carbon. Therefore, it is called as arsenic acid analog. And that was introduced in 1905 by Thomas and Berlin. These two guys used to work in Liverpool, England. So their animal model of testing were mice. They tested this drug on mice. So this atoxyl is based on pentavalent state of arsenic. So later on it was found that the prolonged use of this atoxyl caused severe toxicity in patient. Specifically, it result in blindness due to optic nerve damage, and hence once again there was a need of some effective anti-trypanosomal agent. Sorry, effective anti-trypanosomal agent. So, this quest of finding a new anti-trypanocidal agent, one another molecule was proposed in near future, what we now know it as tripersamide. So this tripersamide was introduced in 1919 by Jacobs and Heidelberger at Rockefeller Institute, and after its discovery, the clinical trials began in 1925 in the Belgian Congo. Okay, so in these clinical trials, it was established that tripersamide was highly effective, and it can be efficiently used to treat late stage sleeping sickness symptoms as such. So by the late 1940s, this tripersamide was widespread as an effective anti-trypanosomasis agent, as well as by the late 40s, treatment failure cases also begin to pop up. So again, there was need to create a new, or you can say to create a more effective, more safe anti-trypanosomal agent. So that gave rise to Millarsoprol era. So what happens? What happened is that by the mid 40s, when the Second World War was at peak, so at the same time, the treatment failure of tripersamide reached greater than 80%. That means whatever patients that were treated with tripersamide for trypanosomasis, of all those patients, 80% of the treatment was failed, and the patient died due to the sleeping sickness. So meanwhile, a young researcher called Ernst Friedhelm, well, he combined British anti-lewisite, what we call it as BAL or dimercaprol, with malarcin oxide to produce millarsoprol. So this Ernst Friedhelm was a British biochemist at Oxford University during Second World War, and he developed this BAL, that is British anti-Lewisite. This BAL was developed as an experimental antidote against the arsenic-based poison gas lewisite and mustard gas that Nazis developed for mass genocide during Second World War. So what Ernst Friedhelm did was that he took that antidote and combined it with Millarcin oxide. As you can see over here, here is the structure of Millarcin oxide, and he combined it with BAL and produce another component called as Millarsoprol, and this formulation is also based on arsenic as its core component. Okay, at that time, this Millarsoprol was called as Melby or Arsobal. Arsobal means arsenic based ball. BAL stand for British anti-lewisite. Arsenic based British Lewis antisite. Lewisite. Okay, so in this way, Millarsoprol was produced. Okay, so now this milarsoprol was introduced for trypanosomiasis treatment in 1949. By 1950, milarsoprol was found to be highly effective. Almost 97% of infection against trypanosamide treatment failure were effectively cured. Milarsoprol somehow is still used today in East Africa as the only effective treatment for advanced stage trypanosomiasis. With time, many treatment failure cases are also recorded now. Okay, so now just imagine whatever treatment therapies that we still use was developed back in 1949, and from 1949 till today 2020, a huge amount of time has been already passed on, and with this time many treatment failure cases are also recorded. So most of the treatment failure case includes what we call it as arsenic in antifreeze effect. So what happens in this effect is that there are convulsions, fever, loss of consciousness. There are rashes on body. The patient might suffer from bloody stools. Patients do suffer from nausea and vomiting. Well, these are the classic symptoms of arsenic in antifreeze effect. So, besides this, 1 to 5% of population that are being treated with this milarsoprol die during treatment, and almost 5 to 10% of case is caused by encephalopathy. So what is encephalopathy? Encephalopathy means any disorder or disease of the brain, especially chronic or degenerative condition that usually hampers the usual, or you can say normal brain functioning. What we call it as encephalopathy. So 5 to 10% of the cases that are being treated by milarsoprol do suffer from such encephalopathy based complications. Okay, now let us talk about what might be the mechanism of action of this milarsoprol. So, to just let you know that the exact mechanism of this milarsoprol is not known. It still remains elusive. So, whatever I am going to tell you is based on the literature that has been accumulated in recent past. Okay, so now what happens is that this milarsoprol is rapidly metabolized in vivo when it comes into the patient's body to other trypanocidal metabolites, and these trypanocidal metabolites can be possibly milarsen oxide. Okay, it can be milanophenyl arsenicals like arsenite and aminophenyl arsine oxide. So what happens is that milarsoprol, once it get inside the body in vivo, it is metabolized into melanophenyl arsenicals, which might include arsenite and aminophenyl arsine oxide. Okay, so all of these trypanosomal metabolites are organic arsenicals and can exist as either with trivalent, or they can exist as in pentavalent state. So to let you know, these pentavalent state are inactive, and hence they still are needed to be converted to trivalent state in order to show its efficacy in vivo conditions. So now these trypanocidal metabolites, whatever they are produced, well, they are hypothesized to get entry into parasite using the parasite specific P2 adenosine transporter, which is expressed by AT1 gene, or they are hypothesized to get entry through aqua glyceroporin 2 transporter, which is a product of AQP2 gene. Okay, so now I am emphasizing on this hypothesized entry of these trypanosomal metabolites into parasite because there are no crystallographic evidence that demonstrate on how exactly these trypanocidal metabolites get entry into parasite cell through either aquaglyceroporin or P2 adenosine transporter. Okay, so the entry is hypothesized through either transport channel, as in case of AQP2 gene, or receptor mediated endocytosis. So the exact uptake mechanism is not uncovered yet. Okay, so now by whatever means, once these toxic metabolites to these trypanosoma parasite are inside the trypanosoma cell, okay, the trypanocidal metabolites form a duct with trypanothione, and this adduct is called as MILT. So these are the trypanocidal metabolites that are formed from millarsoprol. As you can see, there is still few valencies left which are unsatisfied around this arsenic, and these valencies are satisfied by forming a covalent bond with trypanothione's sulfhydryl group. As you can see, these arsenic will form you can say a covalent bond with the sulfhydryl moiety of this trypanothione. So once this melty is formed, which is an adduct of trypanothione and trypanocidal metabolites, so this melty would act as competitive inhibitor of trypanothione reductase. So trypanothione reductase is one of the enzyme that is responsible for maintaining, or you can say for managing, the reactive oxygen species or oxidative damage by the oxidative reactive oxygen species to the parasite as such. Okay, so now this MALT prevents trypanothione reductase dependent antioxidant activity. तो ये मिल्टी क्या करेगा, ट्रिपेनोथायोन रिडक्टेज का जो कुछ भी एंटीऑक्सीडेंट एक्टिविटी है उसको स्टॉप कर देगा. And once the antioxidant activity is stopped, there would be nothing to take care of the oxidative stress that is produced by the host cell, and hence the parasite will die due to oxidative stress. So now in order to demonstrate this theory, what people have done is that they have created the knockdown of trypanothione synthetase and trypanothione reductase, and they figured out that if these enzymes are uh, indeed knocked down, this melty would not show its effect. Okay, so the idea that milty is toxic to cell. Okay, and it requires the presence of trypanothione synthetase and trypanothione reductase. So this is one of the mechanism that is proposed for the action of this millarsoprol. Okay, so now as I have already mentioned, this millarsoprol has been also shown the parasite has been also shown to become resistant to this millarsoprol. So if parasite become resistant to millarsoprol, somehow it also became resistant to pentamidine, and we call this as cross resistance. So if an organism become resistant to one agent, it also acquires resistance to another agent. So such a phenomenon is called as cross resistance. So then work began if the work began if the parasite become resistant to millarsoprol, it also become resistant to pentamidine. We call it as cross resistance. Okay, that is clear for sure. So people started to perform now genome scale screens, and in such genome scale screens, they perturb each gene individually and to ask a question on, like, which genes affected millarsoprol or pentamidine resistance. So this RIT-seq technique, which correspond to RNA interference target sequencing approach took advantage of RNA interference to knock down the gene expression and to ask which knockdown renders parasite drug resistant. Okay, so next, these such RNA, or you can say genome scale screening based on RNA interference technology, well, these studies led them on identification of only one locus in genome that was linked to cross resistance to both the drugs, and this locus encoded two related aquaglyceroborins, AQP2 and AQP3. These AQPs, as I have already told you, codes for aquaglyceroporins. Okay, so now these aquaglyceroporins are among the major intrinsic protein family, and typically facilitate the function, they typically facilitate, or they typically perform the function of passive transport of water and small solute molecules across the membrane. Okay, so now although these findings now suggest, or the association of Millarsoprol resistance being associated with AQP might somehow suggest on, you can say, how drug transport mechanism might have happened. Okay, so as the name suggests, AQPs typically transport only water and small solutes such as glycerol, but the structure of our pentamidine and millarsoprol, it is way too bulkier, it is way too complex as compared to water and glycerol. Okay, so still it don't still it don't match all the dots that we have. Okay, so we still don't know how far larger molecules such as millarsoprol and pentamidine can get entry into the parasite through these AQPs. Okay, so still it is not clearly known. So now to talk about, there are three AQPs. Three AQPs are expressed in trypanosoma: AQP1, AQP2, and AQP3. And of these three, two are associated with the millarsoprol resistance. So this parasite has AQP2 dispensability. This is also one of the most important factor that is needed to be noted. It is dispensable to AQ2P. That means parasite parasite can still live even though AQP2 is defective or if it remains non-functional. Now, AQP1 and AQP3 has got some aromatic arginines that are present in their structures. So, whatever their 3D structure is, they have got some arginine present in the construction from which the water and glycerols are passed, and these arginine are called as aromatic or arginine. And beside this aromatic arginine, there are two selective filter loops. Okay, and this selectivity filter, these one of the loop is called as NPA loop. This NPA stand for N for aspargine, P for proline, and A for alanine. This is one selectivity filter loop, and there is another selectivity filter loop called as NPS. NPS stand for N stand for aspargine, P stand for proline, and S stand for serine. So these along with this aromatic arginine, there are two selectivity filter loops. Those are usually made up of 3 amino acid residue. Due to presence of these selectivity filter, these AQP1 and AQP3, well, they allow only a small solute like water or glycerol to pass through it. But now this AQP2 is very peculiar. Okay, AQP2 do not have that aromatic arginine. That only leads to, that only result, or that selectively leads to pass only water or glycerol through its channel. So these AQP2 lack this aromatic arginine. Well, this means that in absence of aromatic arginine, larger solutes like even our drugs can possibly pass through it. Well, now this is how drugs are hypothesized to pass through AQP2 transport channel. Okay, so now once these parasites sense that such a thing is happening, they acquired mutation in their own AQP2 gene and made it defective so that no more drugs can pass through it, and hence these parasite acquired resistance. Okay, so this is how things are done. So all in all, still there are no crystallographic evidence on how exactly these trypanocidal metabolites get entry into parasite cell through aquaglyceroporins. There are no crystallographic evidence so far, but it is very clear that parasite acquire resistance due to defect in AQP2 gene. Just structural mechanism has to be unveiled or uncovered yet. We know the genetic basis of how these parasite acquire resistance to cross resistance to pentamidine and milarsoprol, but the structural basis is not yet to be solved. Okay, so now it is a good opportunity for you people that you can create a homology model for AQP2 receptor and try to mutate the you can kind of mutate the selectivity filter, or you can mutate that arginine and perform molecular dynamics simulation just to check out what is the change in free energy if such mutations occur. Well, these can give you a computational insights on how these things can happen. Okay, so this was all about your milarsoprol, its mechanism of action, and its resistance. So now the next kind of drug that are not based on arsenic, it includes nifurtimox. Nifurtimox. Okay, so the activity or toxicity of nitrofurans. Nifurtimox is one of the nitrofuran. This is a furan ring, and this is a furan ring. Okay, over here, and you have got nitro group. Therefore, it is called as nitrofuran compound. So, the activity or toxicity of nitrofuran is due to their ability to form free radicals. So, it is this particular functional group that is responsible for the formation, or you can say the toxicity to the trypanosoma as such. Okay, so what happens is that these uh, these nitrofurans, they can form or you can say they can form free radicals that react with molecular oxygen to form superoxide ions. It can form hydrogen peroxide, and it can form hydroxy free radicals. So eventually and collectively, these oxygen containing superoxides, or you can say reactive oxygen species, would result in oxidative stress in the parasite. Okay, so now let us talk about the exact mechanism of action of this nifurtimox. Okay, so this is the structure of your nifurtimox. So what happens is that initially there is a reduction of the nitro group. This is the nitro group that is supposed to be reduced. Okay, so reduced this nitro group is reduced. The reduction of this nitro group from nitrofuran is catalyzed by flavin containing nitroreductase such as NADH or NADPH. So this reduction of this nitrofuran is mediated by this NADH or NADPH. After reduction, it forms something called as the nitroaryl anion free radical. So this is the structure of your nitroaryl anion free radical. So this nitroaryl anion free radical that is formed, it now spontaneously react with the oxygen. Okay, and once it react with oxygen, it leads to the formation of superoxide anion radical. This is called as superoxide anion radical. Once this superoxide anion radical O2 minus is formed, this this nitroaryl anion free radical again attains its original form. Okay, so it gives back the original nitrofuran. So now this dismutation of O2 negative, that is superoxide anion free radical, in presence of superoxide dismutase give rise to hydrogen peroxide. The so form superoxide anion radical, it gets dismuted with the help of enzyme called as superoxide dismutase, which generates hydrogen peroxide. It generates hydrogen peroxide. So this hydrogen peroxide interacts with hydrogen peroxide, again interacts with superoxide anion radical to form hydroxy free radical. Okay, and hydroxy free radical. So collectively, it generates a lot of reactive oxygen species like superoxide anions, hydrogen peroxide, as well as hydroxyl radical. This OH dot and this OH negative stand for hydroxy free radical. So once these hydroxy free radicals get accumulated, that leads to oxidative damage to the proteins, it leads to the oxidative damage to DNA membrane, and eventually the parasite dies. So this is the therapeutics that I needed to discuss with you. So in our next topic, we would be discussing on what are the other therapeutic approaches that can be designed. Okay, so it would be the continuation of our talk that we left at subtractive genomic method of finding out new drug targets, which I explain you taking example of leishmania. While this is a technique, each and every technique has its own merits and demerits. Okay, so I would be explaining the demerits of such subtractive genomic methodologies using the example of this trypanosoma. Okay, we would discuss how these subtractive genomic methodologies that we have discussed in our last few lectures, what are its drawback and what can be done as such. Okay, so I think this is enough for this lecture. Well, let us meet in our next lecture now.